Hi guys, so from today we are going to start a section called the mind maps, right? So we will do mind maps for unit 1 because I know unit 1 gets hard to remember, and now for the people moving on to 12th, 11th is ending and you will start forgetting these names. For students coming up to 11th this year, it can all seem very confusing, too many big words to understand and remember. So let's do mind maps. These mind maps will help you to remember things better and to understand classification in a much easier way. Okay. So biological classification is basically the classification of life forms, right? So we have numerous biodiversity, huge biodiversity, which needed to be classified for easier evaluation, easier studying. For that to happen, for years and generations, humans have tried to classify animals and they have gradually evolved in it. So what did evolution of classification look like? Linnaeus started classifying organisms in two kingdoms, kingdom Plantae and kingdom Animalia. So in Plantae, everything that was green, all fungi, so that had root-like structures, anything resembling roots, as well as bacteria were put into Plantae. And any animal, right, ability of locomotion was put into kingdom Animalia. Quickly, they started to realize that this cannot be the right classification because plants, if green, they are autotrophic, right? Bacteria, they are always unicellular, whereas there are certain animals which can be unicellular. There are fungus which are also unicellular. There are plants which are multicellular. Most fungi are multicellular, right? Again, fungi is not autotrophic, it's heterotrophic. So there was a lot of confusion. And when you group something, for example, when we group kids by their age, all 10-year-olds will be in one group, correct? Now, if we do it on the basis of age and gender, all 10-year-old males and all 10-year-old females will be in two separate groups. You cannot have a concoction of different characters into one group. Then it's not true grouping. So they moved on and they made a third kingdom, which was kingdom Protista. Now in Protista, they added all the unicellular organisms and kept the Plantae and Animalia together. Now what did this was, it separated Euglena. Euglena was a major problem because Euglena is mesotrophic, which means that in the presence of sunlight, it will do autotrophism, but in the absence of sunlight, it will become what? It will become heterotrophic. So this was confusing because where do you put it? Animal or plant, right? But what they at the same time did was they took a few unicellular fungi, unicellular plants, unicellular animals, all of them, and put them into Protista. Now Protista had accumulation of all unicellular organisms, but they could have been plant-like, autotrophic, heterotrophic, right, saprophytic, anything, correct? Then they did a 4-kingdom classification where they took out the Monera, which are the prokaryotes, Protists, which are the unicellulars, Plantae, which are green, multicellular, as well as fungi, and Animalia, heterotrophs. Now see the problem with this classification. The problem with this classification, one is, when they separated Monera out, they realized that all prokaryotes are naturally unicellular. Then what are these? So they have to be unicellular eukaryotes, right? This much was okay. But moving on, Plantae had fungi in it. Now fungi is not autotrophic, but it has a cell wall. If you put it here in Animalia, it is heterotrophic, but it has a cell wall which animals don't have. right? And so then they decided that then fungi cannot belong to either of them. It needs to be separated out, right? So finally, Dr. R.H. Whitaker gave us 5 kingdom classification based on 4 bases of classification. The primary basis of classification was complexity of cell structure. So is the cell prokaryotic or is the cell eukaryotic? To quickly recap, a prokaryotic cell has a cell membrane, cell wall, and it has, I'll not draw it, it has no defined nuclear space. It has no membrane bound organelle. So these were the primitive cells. These were the cells that were formed early on in evolution. These were the cells where the concept of a membrane within a membrane had not yet developed, right? Eukaryotes, on the other hand, were ones with nucleus and other membranous organelles, right? So Whitaker said that all prokaryotes will belong to Monera. Yeah, so that was the first kingdom, kingdom Monera. And all eukaryotes then will be further classified. So this was the first basis. The second basis was this. So all eukaryotes then will further be classified on the basis of number of cells. If they are unicellular, they will become protista. If they are multicellular, then they will be further classified on the basis of their mode of nutrition. Okay, so that means that all prokaryotes are Monera, eukaryotic unicellulars are protista, eukaryotic multicellulars are now getting divided on the classified on the basis of mode of nutrition. So अगर यूकैरियोटिक है और मल्टीसेल्यूलर है, हम चेक करेंगे वो ऑटोट्रॉफ्स है या हेटेरोट्रॉफ्स है. अगर ऑटोट्रॉफिक है मतलब ग्रीन क्लोरोफिल बेयरिंग है, then we will put them in plantae. Okay, and if they are heterotrophic, they can't do photosynthesis or chemosynthesis, photosynthesis actually, then we will classify them on the presence and absence of cell wall. Cell wall is present in all of them. So here, if cell wall is still present, then it's a fungi. If cell wall is absent, then it's animalia, right? So then that is how classification was done of the 5 kingdoms. So you have your unicellular prokaryotic Monera, unicellular eukaryotic. So one step better, eukaryotic still unicellular, protista. Okay, eukaryotic obviously now onwards all will be eukaryotic, multicellular autotrophic, plantae. Okay, multicellular eukaryotic heterotrophic with cell wall, fungi, eukaryotic multicellular heterotrophic without cell wall, plantae. So you have your 5 classifications. Now going on from here, each of these can also be further classified. We will do plant and animal classification in way larger detail. As of now, for the sake of this chapter, I will tell you just the basis of classification for Monera, protista and fungi. Okay. If you want detailed videos on them, you can let me know in the comments and I will try to make them as soon as possible. But as of now, let's focus on the basis. Just on what basis were these classifications done. So bacteria. Bacteria can be classified on the basis of 3 things. One is their shape. If you classify on the basis of shape, then cocci is round, bacilli is rod shaped, Vibrio is comma shaped, and spirillum is spiral. Okay, these are the 4 shape based. Other than that, there can be words like mono, diplo, strepto, staphylo. Okay, so monobacillus or monococcus will mean 1, singular, so it can be monococcus, monobacillus. Diplococcus would be 2, so if I look at coccus, 1, 2. Okay, strepto basically refers to a chain or filament, right? So if it is streptobacillus, then it's a filament of bacillus. If it is Staphylo, it means cluster. So if it is Staphylococcus, then it's a cluster of cocci, like this. So depending on the shape and the arrangement, you can have cocci, bacilli, vibrio spirillum, mono, diplo, strepto, staphylo. Okay. Now, on the basis of staining, you can have gram positive and gram negative. So Dr. Gram is a scientist who did differential staining. Why? Because he saw that bacteria have two kinds of cell wall. One which is very thick with something called peptidoglycan, so it's thick. The other is thin. So what he saw was that if you put a violet colored dye on both the samples, okay, and wash it off, and then put a red colored dye on both the surface, the one that has a thicker membrane, thicker cell wall will retain the violet and therefore the red will not be visible. Whereas the one with the thin cell wall will lose the violet will get washed off and you will be able to see the red. Correct? This is called differential staining, using two stains to see which one holds. So if you see violet, then we call it gram positive, so thicker cell wall. If you see red, it's gram negative. This procedure you do have in detail with a lot of examples. So yes, I will try to cover it in another video in detail with the diagrams as well. Now, moving on to the third type or basis on which you can classify bacteria is kind of their evolutionary time period or their function. So you have archaebacteria. Archae means old, primitive. So these kind of bacteria, they are methanogenic bacteria, they are chemosynthetic bacteria, they can withstand very, very severe temperatures, so they are thermophiles, they can withstand very severe salinity, so they are halophiles. Why? Because these were the forms of life that were produced in primitive earth, and primitive earth as we know it was not as conducive to life or as comfortable as it is now. Temperatures were very high, salt concentration of the ocean water was very, very high. There was no oxygen, there was a lot of methane in the atmosphere, there was a lot of sulfur in the atmosphere. So these bacteria adapted their lives around these circumstances, around these situations, and they evolved to grow such that they are methanogenic in nature, they produce methane, they are halophiles, they like salt, they are thermophiles, they like high temperatures, right? A very good example that you will find in your 12th syllabus is Thermus aquaticus. It's a bacteria which lives in the hot water geysers where water is naturally boiling with geothermal energy. And this bacteria is important because it is now widely used to produce a type of DNA polymerase enzyme which can withstand high temperatures. Right? Temperatures which our normal systemic DNA polymerase cannot survive in, will get destroyed in, and therefore we can use it in artificial biotechnological processes. Okay? Next is U or true bacteria. U bacteria are your present day bacteria. So cyanobacteria, which we also call blue-green algae, it's a misnomer. They are basically organisms which help in photosynthesis. Right? Now moving on, we have Protista. Protista can be classified into a total of 5 subcategories. You have chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. Okay? Protozoans then can further be classified into Coeliophora, those that have cilia, Mastigophora, those that have flagella, Sarcodina, which are amoeboid in shape, and Sporozoa, which are parasitic in nature. Okay? Coming now to fungi. Fungi can be classified into 3 categories. 1, no sexual reproduction. If there is no sexual reproduction, that particular fungus is called fungi imperfecti, and the class or the group is called Deuteromycetes or Deuteromycota. The word mycota means fungus. Then you have true reproduction or sexual reproduction happening. So if it is primitive reproduction, then it's Oomycota, Phycomycetes and Phyomycetes. And if it is advanced sexual reproduction, then it's eumycota, ascomycetes, and basidiomycetes. All of these reproductive cycles you do have in detail, which I will try to upload as soon as I can. If you want detailed videos on each of these subcategories, do let me know. Good luck. Thank you.